I'm Julie Haas. I'm an associate professor in biological sciences here at Lehigh University. My lab is a neuroscience lab, and we study the activity-dependent plasticity of electrical synapses. In the wet lab, we record directly from neurons connected by these synapses. We're looking to understand the conditions and mechanisms that govern plasticity. We also use computational models in order to understand the impact of electrical synapses and plasticity on the thalamic cortical circuit. Electrical synapses are the main form of interneuronal communication within the thalamic reticular nucleus, or the TRN, which is a brain area that acts as an interface between signals coming up from the sensory environment and their target in the cortex. The TRN provides the main inhibitory, or stop, signal along this pathway. In the lab, we record from brain slices that contain the TRN, and we ask how electrical synapses, which are based on the protein connexin 36, change in strength as a result of specific forms of neuronal activity. We use paired whole cell recordings of coupled neurons so that we can measure the synapse between those neurons. We'll show you that next. In this clip, graduate student Mitch is going to bring 2 electrodes down to a brain slice. We've already chosen 2 neurons to target, and now we need to get the electrodes down to where the cells are. The blue traces on the right show electrical signals that tell us how close we are to a neuron. As the line goes up, Mitch is sealing onto a cell membrane. There it goes. The flat lines tell us he's sealed onto 2 neurons at once, and that's what we need to do to record from electrical synapses. Next, Mitch will break into each of the cells with a quick tap, and you'll see the curves that tell us we're listening to the responses of a cell from the inside out. The curves come from the capacitance of the cell membrane. Finally, we'll test how healthy each of these neurons are by looking at voltage responses to current pulses that are each a half a second long and each delivered to one neuron at once. You can see incoming synaptic events here in addition to the voltage responses. And as the current pulses get bigger, we'll see spikes in each of these neurons. We also use computational models of neurons within circuits that contain electrical synapses. We provide our models with inputs that are closely timed, and we ask how electrical synapses in the circuit regulate the information that thalamic neurons pass along to the cortex. I hope this has given you a good idea of what we do in the lab. I'd love to hear from you about what you're interested in doing with us.